Hi everyone, Dr. Hinkey here with our next lecture video. This is on inheritance of traits and patterns of inheritance. So this is our chapter 11. I've posted a chapter 12 video. It's actually from another class, but for right now, till I get caught up here, I will have the rest of the videos by the end of the weekend. But until then, that will give you a way to get the same information. The images will be different. Some of it's directed right toward my microbiology class, but you'll have that information available so you can watch that video and it will cover most of the same material. All right, so our chapter on inheritance, I'm going to start looking at Mendelian inheritance. It's called Mendelian inheritance after Gregor Mendel, who was an Austrian monk. He studied science and math, but he was a monk, so he lived in the monastery. And as one of his duties at the monastery, as a brother there, he conducted breeding experiments. He was in charge of the greenhouses. That was his job. He grew the vegetables for all the monks. And he conducted breeding experiments with the garden pea. And he gathered and documented information from generations and generations of breeding, selective breeding, and what the results were. And what's truly amazing is this was about 100 years before before Watson and Crick discovered DNA. So we knew that whatever was responsible for passing on traits was in the nucleus of cells, but we didn't know what that was. We didn't know how it did that. We didn't understand any of the mechanisms of chromosomes and how they passed traits on. So he figured out patterns of inheritance completely through mathematics. And from that, he formulated the fundamental laws of heredity in the 1860s, 100 years before we knew anything about chromosomes, and he didn't even have a microscope. So this is Gregor Mendel. At the time, the way that scientists communicated, much as they do today, is through publishing their papers. At the time, back in 1860, they didn't necessarily publish in journals, but they wrote letters to the Royal Societies. It was through correspondence and accumulating the correspondence and sharing that with other scientists that information was gathered. Luckily, Mendel shared his findings, shared a lot of his data, not his raw data, but his summaries and his results with other scientists and with the Royal Academy. So we have that. But Gregor was not particularly popular with some of the powers that be. And at the time in the monastery, whoever was the head of the monastery, the abbot, was typically appointed by a king or some political leader. And so when Gregor Mendel passed, the person in charge, the king or whoever the highest political person was, had a falling out with him and wanted to put somebody else in and pretty much erase all signs of any of Mendel's accomplishments. And so he had all of his notebooks, all of his data he had collected over decades burned and destroyed. Lucky for us, that information had already been shared. And once shared and out in the world, it's pretty hard to eliminate factual information. So luckily for us, we do know his findings, and now we know the mechanisms behind his findings. So this is a typical garden pea and the flower. And the fruit and the flower, and we can see we have purple colors. These are the pea pods. We have the peas inside. What was really amazing and allowed Mendel to do this was all of the different characteristics on the pea plant occurred as either or traits. So there were no intermediates. There was no blending of traits, which is incredible dumb luck that he just randomly selected this because as we'll discover later in the chapter, as you've been talking about, many traits, in fact, more traits in humans than not, are not simple Mendelian traits. They don't occur as an either or. We get intermediates, we get combinations, we get overlap, we get multiple genes, we have all sorts of ways that had these traits exhibited some other pattern of inheritance, he would have never been able to formulate these laws. It would have just been too complex. But with the pea plant, luckily, all the observable traits are you either have tall plants or short plants. There are no intermediate height plants. The pods themselves are either inflated or they are constricted, where you can see the indentation around each individual seed. The seeds are either all round or all wrinkled. The seeds are either yellow or green. The flowers on the plant, a plant will either have axial flowers occurring, coming out of the stem where the leaf connects to it, or terminal at the very end of the branch. But a single plant would not have both. A single plant will only have purple flowers, or it will only have white flowers. The pod color will either be green or yellow. So there's not going to be this in-between color. There won't be any in-between both flowers occurring, in-between or combination of purple and pink. So this really allowed him to determine these patterns and the mathematics behind it. Prior to Mendel and prior to him publishing his findings, it was commonly thought that inheritance was this blending. So there are all kinds of different theories. Really, we would call them hypotheses now because they really didn't have any data or evidence supporting them other than people's observations, and that's what we base our hypothesis on. But the common belief, just based on no experimentation to support this with data, was that inheritance was blending. It was a blending of both parents. So if you had parents of contrasting appearance, you would have offspring that were always intermediate in appearance. If we go back even further than this, it gets funnier and funnier. So prior to that, it was believed that all individuals, that in men, in the sperm of men, there were little homunculi, little tiny, perfectly formed humans. And that is what impregnated a woman and grew into the baby. Until you see the blended inheritance. Well, how is that going to happen? How does the man know who he's going to marry and impregnate, or impregnate without marriage? Or how does that happen? Is it predetermined? Is it destiny? But then if the man has a second wife later, why are those children looking a different way than the first one? If those homunculi were preformed, then it became, oh, the woman must carry it. But the same questions. So eventually it came to be, oh, they must be blended. And then Mendel's findings contradicted all of this. And he developed what was called the particulate theory of inheritance. What we would now see as inheritance involving the reshuffling of genes from generation to generation so that we could get any trait from an ancestor. So what Mendel did, those pea plants had all of those many traits I listed, and he looked and isolated one trait at a time for each plant. So he had vast greenhouses at his disposal, and he would segregate out plants based on one specific trait. So he didn't look at purple tall flowers with green inflated pods. He only didn't care what any of those other traits were, only looked at flower color, or only looked at pod shape or pea color, one at a time. And he did crossbreeding experiments. So initially, for any of the traits that he was looking at, he would just cross-pollinate similar plants again and again and again until in the next generation, there was nothing except that trait. So we call that true breeding. He called it true breeding, where if it was purple flowers with purple flowers, they always had purple flowers, and he did that until no white flowers ever popped up in the offspring. We would call those homozygous plants for the flower color. He only chose the varieties of plants that differed in one trait. So he ignored all the other traits, focused on one trait at a time. We call those monohybrid crosses. So mono is a single trait, and a hybrid is something that comes from two different parents. So two different parents that were the same, homozygous for the one trait. And he crossed those and crossed those. Call them reciprocal crosses. And the initial starting generation he selected, that's the P or parental generation for parent, was true breeding. They were always the same for the trait. We call that the parent generation. We breed one parent generation plant time another parent, and we get the first filial generation. So filial is Latin for offspring, for son or daughter. The first filial generation or offspring we call the F1 generation. And then if we select two individuals from the F1 generation, we call that the second filial generation or the F2 generation. So P is the parent, F1 is the first generation, F2 is the second from the parents. Through his observations and recording the data, he formulated the law of segregation of traits. So what he saw in his experiments time after time, and we're going to use flower color, he did this with each of those traits and kept getting the same results. If he crossed those two opposite traits, flowers could either be purple or white. So for generations, he crossbred purple flowers with purple until no white turned up. And he also crossed white with white until no purple showed up. So this was our P or parent generation. When he crossed a purple times a white individual in the P generation, his F1, his first filial generation, had all purple flowers. Then, no matter what trait he was looking at, crossing these true breeding parents, the next generation had all of one trait. And then when he picked any two randomly of those and crossed them, the results always led to an approximate ratio of 3 purple flowers to 1 white. If he crossed any of these F2s with any other F2, it appeared fairly random. Sometimes purple times purple gave all purple. Sometimes purple times purple gave 3 purple and 1 white. But all white times all white always gave white. Purple never showed up when we crossed 2 of the white flowers. From this, mathematically, what Mendel came up with were the laws of segregation of traits. And he summarized it as this: each individual has a pair of factors for each trait. The factors segregate or separate during gamete formation. Each gamete contains only one factor from each pair. Fertilization gives the offspring 2 factors for each trait. So take a minute and look at these. We learned about meiosis in our last chapter, where we have pairs of chromosomes carrying alleles for each trait. During meiosis, we separate out the homologous pairs. Each of those pairs still has its identical twin with it, but because it's identical, it's got the same allele, except for where we may have had crossing over. And then we separate out those sister chromatids. So we divide all of those chromosomes down to a single allele from each pair of homologous chromosomes. We get not homologous chromosomes, but a single duplicated chromosome. And in meiosis 2, we get a single unduplicated chromosome that has one allele for each trait. And at fertilization, we get one allele for each trait. on one chromosome from mom, one chromosome from dad. So if we put that in the vocabulary that you learned in chapter 10, we would say that each trait in a pea plant is controlled by two alleles, the alternate forms of a gene. The dominant allele masks the expression of the recessive allele. Alleles occur on a homologous pair of chromosomes at a particular locus or position on the chromosome. And we use the term homozygous. So the homozygous dominant are identical alleles for the dominant trait. Homozygous recessive are identical alleles for the recessive trait. Or we could be heterozygous, having one of each allele, one dominant and one recessive. So if we look at our homologous chromosomes, here's one from mom, one from dad. And these are the different alleles designated by a capital letter or a small letter. We use the same letter for the same trait. The capital is the dominant. The lowercase is the recessive. We always, when we write our alleles, our genotype, doesn't matter which parent gave us which allele, we would write it as the capital G first. So here, even though, here capital G, lowercase g, here the genotype is capital T, lowercase t. Doesn't matter which parent we got the allele from. During the S phase, these homologous chromosomes, let's call this homologous chromosome number 1, get duplicated. We have sister chromatids. So they have the exact same allele because they're identical. This is my Xerox copy. In meiosis 1, I'm going to separate out the homologous pairs. So these will go to one daughter cell. These sister chromatids will go to one daughter cell. And in meiosis 2, I'm going to separate out the sister chromatids. So this chromosome will go to one of the eggs or sperm, depending on if this is a male or female. Let's say it's a female. This one will go into another egg. This will go into another egg. And this will go into another egg. So here's an example of Mendel's monohybrid cross. This is with tall flowers. So when he took a tall flower, it was pure breeding, so we know it was two capital T's, tall is dominant over short, times the short homozygous recessive with two lowercase t's, because this is recessive. The gametes. Remember, here we start with two chromosomes that are going to be separated. This is a duplicate, so we could write these, but since we're going to have two of each of these and they're going to be separated out, we don't have to. We can just abbreviate to this gamete. This parent could produce eggs with capital T or with capital T. We divide those out. Really what would happen was we'd make a copy of each one and then we'd separate each into 4 gametes. But since these are all the same, we can do a shortcut here and just, this is the only allele we can get in these gametes. Here, this lowercase t, this is the only allele present, so it's the only allele we can get in the gametes. Fertilization, we bring the eggs and the sperm together, and we get the F1 generation that are all tall. But they are heterozygous because they got this lowercase allele for the recessive trait from this parent. 100% of my F1 generation have the dominant allele and the recessive allele, the capital and lowercase. We pick any two of these individuals and we cross them. And now what the gametes are going to be, well, during the S phase, I'm going to make exact copies of this, so I'll have 2 capital T's and 2 lowercase t's. Then I'm going to separate all of those out into possible gametes. So let's say pollen, in this case, sperm. I could have 2 capital T's or 2 small t's, either one of those, because 2 capital T's, both of those are going to give me the same results. two small t's, both of those are going to give me the same result. I only have to divide this into capital T, lowercase t are my possibilities in the eggs. Capital T or lowercase t are my possible alleles for this trait in the sperm or in the pollen. So meiosis, we go from 2N to N. That 2N is right here. I have these two alleles. I have these two alleles. In my gamete, I only have one. Could be either one of those, but since they're the same, that from 2N to N. Fertilization, N plus N, 2N. Here I have my 2N. When I divide 2N to half of these, I could have either a capital T or a small. So here I need to represent both of those in my Punnett squares. Fertilization, I bring this. These are all my possible outcomes. I don't know which one I'm going to get. These are all the possible outcomes. Two capital T's for a homozygous dominant tall plant, a capital T and a lowercase t for a heterozygous tall plant, another capital T, but now from the opposite parent, lowercase t from opposite parent, heterozygous tall plant, and a lowercase t from one parent and lowercase t from the other gives me the homozygote recessive. If I count up the possible genotypes, I have homozygous dominant, 1 out of 4, heterozygotes, 2 out of 4, or 1/2, and homozygous recessive, 1 out of 4. So those represent my genotype ratios. I can write it as the number of squares out of the total number, 1 out of 4, as 25%, or as 1 with the colon, 2 of this one, another colon, and 1 of this. So my genotype ratio is 1 to 2 to 1, or 25% homozygous dominant, 50% heterozygote, 25% homozygous recessive. My phenotype ratio, when I have the dominant allele, it masks the expression of this lowercase allele. I don't see that small t. I don't see a short gene. It masks its expression. It wins. And all of these are tall plants. So phenotype ratio is 3 to 1 short. 3 tall to 1 short, or 75% tall, 1 short. So genotype versus phenotype. Genotype refers to the two different alleles an individual has for a specific trait. It does not refer to what the individual looks like. If I have two identical alleles, then I have a homozygous genotype. If they are different, they are heterozygote. If they're homozygous, I have to say which one, homozygous dominant or recessive. If they're heterozygote, it tells me this hetero different and this zygote, two different alleles. So I don't have to put a dominant or recessive after that. That's just heterozygous already tells me what it is. My phenotype refers to the physical appearance of the individual. I have to give the qualitative description, tall, short. Right? So phenotype is not dominant recessive. That's what I use to tell which of the homozygosis I have in my genotype. The phenotype is the actual description, tall, short. So my Punnett square is a table that lists all the possible genotypes resulting from a cross. I don't know which one I'm going to get, but this tells me the odds of getting any one of them anytime I have a breeding event. So all the possible sperm genotypes are lined up on one side, let's say across the top for the columns, and all the possible egg genotypes are lined up on the other side, so down the rows to bring them together. It doesn't matter which one goes where. Sperm can be across the top, eggs on the side, so columns and rows, or they can be reversed. It doesn't matter as long as the same gamete is in the same orientation. And then every possible zygote from these genotypes, or every possible genotype of the zygotes, are placed within the squares by bringing the two together. So here's an example of a human trait with unattached earlobes and attached earlobes. We have a key telling us the unattached earlobes are the dominant trait. They will mask expression of the lowercase e attached earlobes. In this case, my parent generation have two parents with unattached earlobes, and we're told they're both the heterozygote. This parent can give me either a capital E or a lowercase e for gametes, 2n to n or n. 2n here is a capital E lowercase e. To go to n, I split those up, e capital or e lowercase. And then in my Punnett square, I bring that column and row together, Notice it doesn't matter which parent I get this from. The capital or dominant allele always comes first, is written first, just by convention. And here we can see again, heterozygote for two of them has the same phenotype as homozygote dominant. So I have 3-to-1 ratio of the dominant phenotype of unattached earlobes. So 3 unattached earlobes to every 1 attached earlobes, or 75% of the offspring. You have a 75% chance of getting offspring with unattached earlobes. Or if I wanted to talk about genotype, it would be 25% homozygous dominant, 50% heterozygote, and 25% homozygous recessive. We kind of did this in our discussion already, so we're not going to do that. We looked at your individual traits. So something that Mendel was able to come up with based on his findings was a way to determine, if I don't know, I can't tell by looking, so I can't tell by the phenotype what the genotype is if I have the dominant trait. So I can't tell if I have a purple flower just by looking at that purple flower. I don't know if it's homozygous dominant or if it's heterozygous. But I do know the genotype for the homozygous recessive trait, because the only way to express the homozygous recessive trait, the only way we see that is with 2 lowercase homozygous recessive alleles. So if I want to figure out what the genotype of a dominant trait phenotype is, what Mendel determined was, if I cross it with a known genotype, so my recessive, then I can determine what the individual alleles are in that parent with the dominant phenotype. So if I have the dominant phenotype, I don't know if I'm, let's say, capital P for purple flowers or capital P lowercase p for heterozygous purple flowers. So it could be either one of those. So take that unknown and cross it with what you do know, which would be a homozygous recessive. So for our tall and short plants, if we had a tall plant, we have no idea what this genotype is, but we do know, we do know that they do have to have at least one capital T, right? So we know at least one of these has to be a capital T for this to be a tall plant. We know for sure both alleles here. So we know the only allele coming from this plant is this lowercase t. Here, I don't know, but when I do the test cross, depending on what I get here, determines it. I know there's one capital T because this is tall. So I know this is capital T. I know these plants will be tall. And I know this gave a recessive allele, so it has to be this lowercase t. All right, so to be, if I came up with a tall plant, it could have been either one of those, but I know it's not this because this parent only had lowercase alleles to provide. This is a short plant, so I know that this parent had to also provide a lowercase recessive allele. So I can figure that out by working backwards. So here, in this case, I don't know what my phenotype is here. I do know the only allele I have here is T. If 100% of the offspring are tall, then I know both of the alleles presented from this one have to be capital T because I got no short offspring. All right, we can really muck with things now and look at how about two traits? How would two traits be passed on? What if I wanted to know how are tall plant alleles and what are my odds of having a tall plant with purple flowers? This could get much more complicated. I might want to know what are my odds of having a tall plant with purple flowers and inflated pea pods. So I could do 3 trait crosses. That's where computers come in handy because it gets complicated. We're only going to go to a dihybrid cross. What a dihybrid cross is, 2 trait inheritance. We're looking at true breeding plants for 2 different traits. So take the tall plant with purple flowers and keep taking tall plants with purple flowers and crossbreeding them until the only outcome are tall plants with purple flowers. I never get a tall plant with a white flower or a short plant with a white flower or a short plant with a purple flower. It's always tall plants, purple flowers, and then short plants, white flowers. I'm going to keep breeding until there's nothing else that ever comes up but short plants, white flowers. So 2 traits together on the same plant. And then do that same process. Cross those would be the P generation, the parent generation. Cross those 2 true breeding plants. So tall purple flowers, short white flowers. That's my parent generation. Cross those. 100% of the offspring, that F1 generation, are going to be tall plants with purple flowers because those are the 2 dominant traits. I take 2 of those F1 plants, the heterozygote for both traits, and I cross them and see what phenotypes I get in the F2 plants. From the results, which we'll get to in a minute, Mendel came up with the law of independent assortment. We talked about independent assortment during meiosis. Without knowing chromosomes or alleles or genes, what Mendel said was the pair of factors for one trait segregate independently of the factors for other traits. So that all possible combinations of factors are Mendel speak for alleles, can occur in the gametes. I can get any combination. So just because I had tall purple plants and short white plants in my P generation, and then all tall purple in my second, when I crossed those, I'm not only going to see tall with purple and short with white. I'm going to get tall with white and short with purple, any combination. So independent assortment, if we look at independent assortment and segregation during meiosis, say here are my chromosomes that I got from mom, that I got from dad. So these are a homologous pair, and these are a homologous pair, I have different alleles in each one. So this would be my heterozygote F1 generation. The parent cell has 2 pairs of homologous chromosomes with 2 traits, trait A and trait B. So during the S phase, we are going to duplicate each of these, get a sister chromatid, but we're going to still have the same alleles on those sisters because it's an exact copy. Right. And then they are going to line up at the metaphase plate. This chromosome lines up with its homologue. This chromosome lines up with this homologue. They carry genes for the same trait at the same locus. They don't necessarily carry the same allele. They can have alternatives of that same trait. So the alternative is capital A lowercase, capital B lowercase. They can line up because of independent assortment. I could have them line up with this one from mom and this one from dad on this side. So those two would go together in a gamete, and these two would go in together in a gamete. Or I could have them line up with both of these on one side, both of these on the other. Or I could switch this, or I could reverse those. So all of my possible outcomes for the gametes after we go through meiosis 1, so I've separated out the homologues. I could have this combination and this combination, and this combination and this combination. So at metaphase, each daughter cell has 1 member of each homologous pair. So we're in meiosis 2 now, right? The sister chromatids have lined up and are separated in meiosis 2. Have all possible combinations. A capital A with a capital B, a lowercase a with a lowercase b, a capital A with a lowercase b, or lowercase a with a capital B. So that independent assortment, these lining up differently, this is what gives us this variation. This is without crossing over, which could switch and swap some other genes. All right. So when I do a 2-trait dihybrid cross, I start with pure breeding. Pure breeding homozygous dominant for both traits. So here I have tall with a green pea pod. Here I have homozygous recessive, short with a yellow pea pod. When I divide these, this is my 2n number. I have 2 T's, 2 G's. That's my 2n number. When I go to n, I end up with half the number of chromosomes. I have half the number of alleles. We'll have 2 alleles for one trait, 2 alleles for the other, 4 total. 2n to n, I go to 2, right? So here I have 1, 2, 3, 4 alleles. I'm going to go to 2 alleles, 1 for each trait. So I have to have 2 different, I can't get 2 T's. That would be like getting 2 of the same chromosome in a gamete, which would be a birth defect like trisomy 21, right? I have to only get 1 of each of these, but I do get 1 of each. So 1 T, 1 G. What are the possible ways I can get 1 T with 1 G? Well, the only T I have are capitals. So no matter what, I get a capital T. Here, no matter what, I get a capital G. So when I have the homozygote recessives, I know my gametes really easily. Here, I only have lowercase t's and lowercase g's, so I know all my gametes contain lowercase tg. Fertilization brings those together in the F1. I get heterozygote, this T with this T, gives me the heterozygote tall. The capital G with the lowercase g give me heterozygote green pea pods in the F1 generation. Now, a little trickier to figure out the gametes. what are all the possible ways I can combine one of these T's with one of these G's? Well, I can get this capital T with this capital G. There it is. Or I can get the capital T with the lowercase g. So I can work with one of the same letter at a time. So I have two T's. How can I do this one T? All right, how can I divide out this other T with one G? Got the dog alarm going off now because Amazon's here. So let's pause for a second. Okay, I think the dog alarm's done. All right, so how can I divide this lowercase t with one of these g's? I can do lowercase t with a capital G, lowercase t with a lowercase g. Those are all of my possible gametes. From 2n is 4 to n is 2, one allele for each trait. Since I'm crossing two of the same genotypes, I have the same alleles on this axis, and I bring these two axes together. Capital T and a capital T gives me two capital T's. Capital G gives me two G's. Again, we're going to keep the same trait together when we have fertilization. So I have two alleles for this trait, so I put my same trait alleles together. I wouldn't write TG. So capital T, capital G. Bring all of these together. Then you have to figure out what are the genotypes and phenotypes. So genotype, just see how many of these do I have? Well, with two capital G's, homozygous dominant for both traits, I have one. Homozygote recessive for both traits, I have one. Homozygous for one, heterozygote for the other. For tall, heterozygote for green, I've got those two there. I'd have to do all that counting. It ends up being 1, 2, 1, 2, 1, 2. Oh, wait, there's a 4 in there. These 4. 1, 2, 1, 2, 4, 2, 1, 2, 1. So I could count those. I would have to go through and count. Phenotype's a little bit easier. Can look what are my 4 possible combinations? Stop! Hey! So my possible combinations with tall and green or short and yellow are tall and green, tall and yellow, short and green, short and yellow. Those are my phenotypes. And when I add those up, my ratio is 9 of the plants out of the 16. I have 16 squares when I put a 4x4 grid together. 9 show the dominant phenotype for both traits. 3 show the dominant for one trait and the recessive for the other. Another 3 do the reverse of that, the recessive for the first trait, the dominant for the second. And 1 shows both of those. So just like with the monohybrid cross, our F2 generation phenotype ratio was always 3 to 1 if we crossed pure breeding in the parent, or if we crossed the heterozygotes, we always got 3 to 1. With a dihybrid cross, if the parents are pure breeding, the F1 is always 100% for the heterozygote. When we cross the heterozygotes, we get 16 possible outcomes, always with a 9 to 3 to 3 to 1 phenotype ratio. So we can use that to look at inheritance patterns in humans. We've done that for just regular traits. We can also track and predict the potential for inheriting a genetic disorder. So the autosomes, if we talked about, are any chromosome other than the sex chromosomes, so numbers 1 through 23. Genetic disorders that are caused by genes on chromosomes 1 through 22 are called autosomal disorders. And we can look at the pattern of inheritance, and sometimes we see that these autosomal disorders are dominant. We call these autosomal dominant disorders. Autosomal dominant disorders are characterized by being present with either the homozygous dominant or the heterozygote. All you have to do is have one dominant allele to have the disorder. The only way you don't inherit the disorder is if you get two recessive alleles from your parent. There are other genetic disorders that pass on the trait as autosomal recessive. So if you have two dominant alleles, you're homozygous dominant, you do not have the disorder. If you are the heterozygote, you do not have the disorder. You carry it, you carry one allele for it, but because this capital dominant is for the normal expression of the gene, you do not express the disorder. The only way you do have the disorder is if you have the homozygous recessive. Right? So the name tells you autosomal dominant means if I have the dominant allele, I get the disorder. Autosomal recessive tells me I have to have the recessive twice because the dominant always masks the recessive. So the only way to get it is by having the recessive allele twice. I can do Punnett squares would allow me to predict what are the chances that an offspring will have the disorder. I use pedigree charts to determine phenotypes within a family to look at what the inheritance pattern is to help me determine what are the odds that an offspring will carry it. Well, I have to know what the inheritance pattern is. So in a pedigree, square boxes represent males, circles represent females. If it is shaded in, it means you are affected. You have the disorder. If it is not shaded in, you do not have the disorder. So we can use this combination to figure out what's the inheritance pattern, and I can go back and figure out what these unknowns are. So remember for all of these characteristics for Mendelian inheritance, the heterozygote and the homozygote dominant have the same phenotype. So I can't differentiate those two. So I can use this, looking at the pattern of inheritance, to figure out what these unknowns are. So if I look at an autosomal recessive pedigree, here I see, well, females seem to be affected more than males. And not a lot of individuals are affected. Seems to skip a generation, doesn't it? Or two, married relatives here. But it doesn't show up frequently. So here, this is indicative of an autosomal recessive disorder. I look at the chart and I say most affected children have unaffected parents. The only way I can have an offspring that's affected, but parents that aren't, is through the heterozygote. Because if this was autosomal dominant, then anybody carrying this dominant allele would be affected. So we have to have, in order to have two unaffected parents have offspring that do have this, they both have to be carriers. They both have to be the heterozygote. So this skipping a generation and parents that aren't affected having offspring that are, or a parent that is affected with one that's not having offspring that are not affected, is indicative of an autosomal recessive pedigree chart. So two affected parents will always have affected children. I don't see that here. But if I did see that, it would tell me definitely. Affected individuals that have a homozygous unaffected mate will have unaffected children. So we see this here. This is unaffected, it's not shaded in, and all of their children do not exhibit the trait. Close relatives who reproduce are more likely to have affected children. And we see this here because both of these, all of the offspring here had to get an allele for the affected trait because that's the only one that mom carries. We don't know what dad's second one is, but if it was affected, then we would expect some of these offspring to be affected and none are. So we can assume this is a dominant allele. But we know all the children have this lowercase allele that mom gave them. In this case, we have these offspring, none are affected, but when two cousins interbreed, then those two recessive alleles that they each inherited from grandma come back together. Typically with this, we see both males and females affected equally. Well, here, yeah, we do have, not equally, but we do have both males and females affected, even though there's two females. But both of them, we see either sex affected. For autosomal dominant pedigree, the first thing that should jump out at you is how many individuals have this. So lots of affected individuals, both male and female. Affected children will usually have affected parents. Heterozygotes are affected, so we see it frequently because the heterozygote's not protecting them. Having the dominant allele is what causes it. It doesn't protect it, and it masks the recessive or the healthy allele. So two unaffected parents, anytime we have unaffected parents, we know they must have the recessive alleles. Two unaffected parents will never have children with the disorder. But any affected parent will pass on that allele and will always have children that have, will always have some children that have that disorder. So those are what we look for to determine an autosomal dominant inheritance pattern. And then we can look back and forth, work back and forth to figure out what other traits are based on, well, if we didn't know what this parent was, we only knew they were affected, we'd know they would have to have a capital. Right? This is lowercase, so we would know they're a heterozygote because we have an offspring that's not affected here. So dad has to be able to give one of the unaffected alleles to have any children that are unaffected. So some examples of autosomal recessive disorders. You think back to our research into enzymes and some of the disorders you looked up. I think some of you looked up Tay-Sachs disease. It's progressive deterioration of psychomotor functions due to the lack of that one enzyme to break down a lipid to do its job. So that was the inheritance patterns. You noticed those inheritance patterns and wondered about those. Cystic fibrosis is a disorder that results in mucus in the bronchial tubes. It builds up and in the pancreatic ducts. It's very, very thick and viscous. The plasma membrane channel for the chloride ion doesn't function properly, and so we don't maintain water balance, leading to that very thick mucus. Methemoglobinemia, or the blue fugates. If you look that up, you'll see. This is an alternate. They lack an enzyme that converts hemoglobin intermediate, methemoglobin, back into hemoglobin. And so it ends up, it never gets that red color. So circulating under the skin, it always seems to have a bluish tinge. There are some colorized photos showing the blue fugates, this family in the Appalachians looking very blue. It's just really, really pale, slightly blue in real life, not as blue as some of the images, but you can Google that to see those. I think there might be a picture in the book too. Some examples of autosomal dominant disorders. Osteogenesis imperfecta, Mr. Glass, if you're a comic book fan. This is called brittle bone disease. It's a mutation in two genes that are needed to synthesize collagen, which is what gives our bones their strength and order. It has to be the right shape in order to build those firm bones, and it's not, and so bones break easily. Huntington disease is a neurological disorder, causes progressive degeneration of brain cells. You get personality disorders, severe muscle spasms, a lot of nervous system problems with that. And it's autosomal dominant. Why would autosomal dominant disorders stay? Why would you pass those on? One of the heartbreaking things about Huntington disease, for example, is that it does not show up until you are in your mid to late 40s. So by that time, you have probably already had children of your own before you even realize that you are affected. And so you would pass it on without even knowing that you were able to pass it on. All right, so some patterns of inheritance that aren't just simple dominant recessive. Right? They're a little more complicated or a little bit different, would have really messed up the math for Mendel. Incomplete dominance is when the heterozygote has a phenotype that's intermediate. It's a halfway, it's a blending. This is our blended inheritance. So there's some traits that it doesn't end up with the homozygote or heterozygote, or homozygote, recessive, or dominant. It ends up an in-between. So an example would be a flower that has a homozygous red, gives a red phenotype, homozygous white gives a white, rather than homozygous, the heterozygote being red because it has a dominant allele. We would end up with a blending of the white and the pink phenotypes, and the heterozygote would give us pink, an intermediate. So incomplete dominance is a blending. The phenotype tells us what the genotype is without a test cross. We can just look at it and say it's pink, it must be the heterozygote. So sickle cell, which we'll talk about a little bit more, and wavy hair are examples of this in humans. Here's a red and white flower giving us pink. Rather than having that 3-to-1 ratio after we do a pure cross and then get the heterozygote and cross the heterozygote, crossing the heterozygote gives us a 1-to-1 ratio because the heterozygote doesn't look like the homozygote's dominant in this. So we have a distinct heterozygote phenotype. We have some traits that are multiple allelic, meaning we don't just have capital and lowercase alleles, we have multiple alleles. So we have more than 2 possible outcomes. So the gene exists in several different forms. Each individual still only gets 2 alleles, but it's more than just a capital and a lowercase. So an example is ABO blood types. Our possible alleles, the letter A, B, or O, that A indicates that you have the A antigen on your red cells. Right? So you have anti-B antibody in your plasma. This means your antibodies, the things in your immune system that attack foreign bodies, attack B proteins on blood cell surfaces, or B antigens on the surface of your red blood cells. Right? So you have the A antigen. If you had a blood cell with B on it, it would get attacked. So you are A blood type. Because there are multiple alleles, we write this as I. The I for here is for immunoglobulin because we're looking at antibodies. And then the superscript A. We do this is a way to indicate that, oh, there's something different about this. It's not just that I'm looking at 2 of the same letters, capital and lowercase. I'm looking at more than that for combinations. So if I have the B antigen on my red blood cells, I am blood type B. I have anti-A antibodies. Sorry, that should be an anti-A antibody. Anti-A antibody. There, fix that for you. So I am a blood type B. because my antibodies, my immune system will attack A antigens. And then if I have, look at all the typos in this, lowercase I is indicative that I have neither one of those. So the lowercase tells me I have neither one of those alleles. I get two alleles. I have to have two alleles for every trait. So these are single alleles. The combinations that I can have two alleles for blood type A. This actually works out if you write it this way, but so you can solve problems this way. This doesn't work with the sex linked, but for this one you can. But the proper way to write that would be, I can be the homozygote dominant for A, or I can be heterozygote for A, and I have no other antigens. So what this signifies is, remember those surface proteins on my blood cells or on all my body cells? I have these proteins that are markers A or B. If I am blood type A, and some people have A markers, some people have B markers, some people don't get any of those markers. There is nothing, they don't have A or B antigens, right? So they have antibodies against both A and B. So when we call that O, the O is not the letter O, it's actually 0, for I don't have any of these antibodies. I don't have any of these antigens on my on my cell surface. So that's what the O actually stands for. So I can be type A if I have the homozygote dominant. If I have homozygote A for A, I have two A alleles. That means all of my cells have lots and lots of antigen A. I can be the heterozygote with O. So A, O, meaning I still have A on my cell surfaces, but I might just have half as many. I can be B, type B, as either a homozygote for it, meaning I have lots of B antigen on my red cells, or B with the I, with none. So this is also, beside multiple allelic, this is also codominance. Both alleles are expressed. So if I have two A alleles, I have lots of A on the surface. If I have one A and one O, I have half of the A, and then, I only have half because that doesn't produce any. Same thing with the B. If I get both, one A and one B, I am blood type AB, right? And don't have any antibodies in my blood. Or I can be type O, which I'd have two alleles. I have two of the same alleles. Blood type O is both of these lower cases I don't have A or B antigen. I have both types of antibody. So I can actually do, for this, I can do my Punnett square using these as my possible blood types. This is the correct way to do it, just to indicate that, A and B don't mean two different traits. Typically, for a trait, I have the same letter. This lets me know this is what's different about this, is I can have different letters, different letter combinations in my alleles. So when I do this, I would figure out what the blood type is. Here I have a heterozygote blood type B male and a blood type A heterozygote female, so AO, BO. I would separate out those alleles. Here's my B allele. There's my blood type O allele. There's my blood type A allele. And there's my blood type O allele. And I bring those together. I have a 25% chance of having offspring with blood type AB, 25% chance of having offspring with blood type B, but the heterozygote, 25% chance of having blood type A, the heterozygote, and a 25% chance of having blood type O. Some other non-Mendelian inheritance, polygenic inheritance, occurs when I have a trait that is governed by more than one gene. Lots of different genes are going to interact. So some of those have a dominant allele. All the dominant alleles would have a quantitative effect on the phenotype. We're going to add these up. They're additive. But some of these genes, for example, eye color always comes up because people say, oh, I can't have blue eyes. Nobody in my family has blue eyes. Eye color's regulated by 15 different genes that have been identified so far, maybe more. So if you get the right combination of recessive alleles from two sides of the family, pretty much any color can pop up. So it's not as easy to figure out with polygenic traits. But what we find is continuous variation of phenotypes. So anything that if I chart out among the population and get a bell curve in variation, then it's probably polygenic. So eye color can be very, very light all the way to very, very dark, and the majority of individuals are somewhere in the middle. Skin color, height, IQ, seed color, and wheat. These are all examples of polygenic traits. Right? If I plot out height in a population, I get this bell-shaped curve. Here I could do that as females, males, but mostly a bell shape within the population. This would be my distribution from multiple alleles. Here they're giving an example with genotypes for a trihybrid cross for 3 different alleles and the different combinations, the results those would give you. Pleiotropy is when one gene affects more than one characteristic of an individual. So sickle cell anemia is an example of incomplete dominance, but it's also pleiotropic. It's a single gene but has lots and lots of different effects on an individual. Codominance is when more than one allele is fully expressed. So ABO blood type is an example. If I have A and B, both are expressed. I get both proteins on the cell surface. Some flowers, if I get red and white, I don't get pink. I don't get red, the dominant. I get both red and pink on the petal. A lot of animal coats are that way, both colors. I cross a black and a white parent, and I get black and white in the offspring. So both are expressed. Epistasis is when a gene at one site interferes with the expression of a different gene. So albinism is an example of this, where albinism isn't due to a mutation at the gene that's actually producing pigment. It's a different gene, but it blocks expression of the gene producing pigment, results in albinism. Can also have some environmental effects in how genes are expressed. So I might have the gene that produces a specific enzyme, and the enzyme is what's responsible for giving me the traits, but as you learned in lab, enzyme function is affected by temperature, pH, salinity, all those other factors, and many enzymes either don't work or they do work only at specific temperatures. So an example, the points in Siamese cats, that enzyme is only active at colder temperatures that lays down the pigment in those places, and that's why you only get color at the extremities because those are going to be a little colder. Arctic fox coat color depends on their temperature. The Himalayan rabbits are the example that they give you in the book. Another example is sex-linked traits. Sex-linked traits we have, if we have no copy of the normal allele, so this here we have a sex-linked trait for hemophilia, is the example that I think you worked on in lab. So if you have no copy of a normal allele, then your phenotype is that you have this clotting disorder. Right? So one copy of the normal allele and you're okay. But this is carried on the X chromosome. If you remember from our karyotypes, the X chromosome is very long, the Y chromosome is short. X-linked traits are governed by genes on that portion of the X chromosome that there's no corresponding Y, right? That those genes have been lost on the Y chromosome. They're not there, Right? So whatever you get on the X chromosome is what you get. Right? So these are carried on the X chromosome, and we don't have a balance. We don't have a second allele on the Y chromosome because that portion of the Y chromosome is missing. We've got a little short, little dinky Y chromosome. So there are locuses, positions on the X chromosome that don't have a corresponding part on Y. So that means we always have to write X and Y because what alleles we get are going to be different from males to females. So we have to know if we're talking about a male, so we have an X and a Y, or are we talking about a female? So we have two X's. So for this case, we always have to tell first what our chromosomes are, X and Y for dad, two X's for mom, and then what allele they carry for the trait. So we superscript that. The normal allele, not affected, is the capital letter in this case, because this is a recessive trait. Same with color blindness. Dad's genotypes can be, dad is always XY. His X chromosome can carry the normal allele, the capital H, or it can carry the lowercase h. The Y doesn't have a corresponding part. Dad has a Y chromosome. I show that here because when I do my Punnett square, I have to know what alleles can be passed on. Well, if I get the X chromosome, this could be passed on. If I get the Y chromosome, there's no allele. So have to show both of these, right, to show how this is inherited. But dad only has two possible outcomes because the Y doesn't carry anything. So he either gets the normal allele from his mother or he gets an affected allele from his mother. Mom has two X chromosomes. So she is going to have two alleles. She can be homozygote dominant, meaning not affected at all, normal. She can be the heterozygote. She will still not have the disorder, but she is a carrier of the disorder, the lowercase. Or she could be affected. For a female to be affected, they must carry both of the alleles for the disorder. So both of the lowercase h's for hemophilia. That means daughters can be protected even if they get the allele. They can be protected as long as the other parent gave them a normal allele. Males get what they get from mom. They have a 50-50 chance if mom is affected or a carrier, or if mom is a carrier. If mom's affected, that's all they get. So color blindness is an example, and hemophilia is an example. If you always write your alleles, your genotypes like this, with the X and the Y, and then the superscript for your allele, and then divide these out by what chromosome they get from each parent in the Punnett square, you'll get the correct results. We also have to know this because how the offspring are affected is different depending on if you're a male or a female because of this. So just remember, here are my possible genotypes for dad in the Punnett square. He could pass on X, affected by hemophilia here. He will pass that on to daughters because mom's only giving X, so any offspring that gets this will be a daughter. Dad only passes on Y to sons. Dad can't pass this on to any sons because the Y doesn't carry it. Sons are going to get this through whatever X they get from mom. All right, the last thing I want to mention on this is, why would something harmful stay in the population? So I mentioned with Huntington disease, it's because you don't know you have it until you are past reproductive age, and probably have passed it on already without knowing it. Sickle cell disease is an example of something that is harmful. It stays in the gene pool. For sickle cell anemia, it is incomplete dominance. If you have two alleles for sickle cell, so if you have two normal alleles, let's say two capital S alleles, you are normal. You do not have sickle cell. You are healthy. Your hemoglobin is correctly shapen. It flows well. It carries lots of oxygen. If you are homozygote recessive and have two alleles for sickle cell, your hemoglobin is misshapen and it cannot carry oxygen very well. That leads to your red blood cells being sickle-shaped. They do not flow very well. They tend to aggregate and clot, and you have circulatory problems and a whole host of other problems related to a lack of oxygen, circulation, and clotting. The heterozygote, because this is incomplete, because it's co-dominance, has some blood cells that are properly, the right shape. The hemoglobin is functional, everything works, and some that are not. So they're basically somewhere in, they have half of the adverse impacts to their health. So they are relatively healthy. Still, they do have some adverse impacts. People with sickle cell tend to have have so many health problems that they reproduce at a lower rate. They often do not survive well into reproductive years, so don't have so many children to pass on. Eventually, something that is harmful like that would be eliminated from the population. But in this case, sickle cell has stayed around in about the same percentage in the population for hundreds of years. The reason is in areas with malaria, so tropical and subtropical regions with the Aedes aegypti mosquito, with the mosquito that carries Plasmodium, the parasite that's responsible for malaria. In areas where malaria is present, people with sickled cells, the Plasmodium, that parasite, matures in red blood cells. If you have sickled cells, it can't enter. It does not reproduce, the larvae don't grow in your sickled cells. So you are protected. You never get malaria. If you have normal homozygote dominant, normal red blood cells, normal hemoglobin, chances are you're going to catch malaria and die if you live in those regions. If you are the heterozygote, however, you have some adverse health impacts, but not nearly as many. You'll probably survive and be relatively healthy. You'll have some minor impacts, but you can survive into old age and you can have offspring and you can live a pretty normal, healthy life. And because you have some sickled cells, because you do have some of the misshapen hemoglobin, you are protected from malaria. In this case, we call that the heterozygote advantage. It can keep harmful alleles in the population because in the heterozygote, you have some advantage over either of the homozygotes, and that's why that stays in the population. All right, so that is it for this chapter. I hope that helped, and I will get the rest of the videos up this weekend.